Thanks for the opportunity to be here. During our brief introduction when we went around the room, I mentioned that I was excited about this. I'm sort of semi-retired. I don't go to many meetings anymore. I'm rather selective. And when the opportunity to come here came up, I was excited precisely because we have now people with analytical math and modeling skills, statistical skills, and people with practical experience in the industry who have some insight into what the actual problems and enigmas are. And I hope that together we can make real progress on this. I'm going to spend a couple of minutes following up on what Jim Brennan talked about. He talked about contamination in the field during the growing process, some of the issues associated with that, and I'd like to talk about the next step, which is post-harvest food safety, what happens after we've harvested those commodities and the further handling. Some of this presentation is really a repeat of a presentation I made last year for the Center for Produce Safety. So Callie, you can take a vacation if you'd like. You've heard some of this. Some in the room have heard some of it. Hopefully some of it will be new and different. I think the first thing to be cognizant of is that the life cycles and the ways that these pathogens get around are variable. And there are certain pathogens that are most likely to contaminate fruits and vegetables in the field in the growing process. And there are other pathogens that are perhaps more likely to contaminate our products post-harvest or during harvest. And the reason is simple. There are a number of pathogens that are typically associated with human beings. The viruses, for the most part, are specific to human beings. They don't have animal reservoirs. And so if we get contamination with norovirus or hepatitis or the like, it came from human handling. And most human handling of these products occurs post-harvest, not while they're in the fields. Similarly, Staph aureus, which produces a toxin, typically becomes a problem when you are handling a product. You've handled, your fingers have gone in your mouth, your nose, which is the main reservoir for this bacterium, and then the bacterium can grow on the product, produce the toxin, and make people sick. This is less likely to happen in the field. It's more likely to happen in a handling facility, a restaurant, a picnic, in your home, and the like. So these are the kinds of things that are going to contaminate our products post-harvest. And of course, they're important issues, and there have been some major outbreaks. Other contaminants, such as the EHEC, E. coli, salmonella, things like this. Again, there can be human reservoirs. You can have people in your handling facilities that are suffering from salmonellosis and could potentially contaminate the products. But for the most part, my expectation would be these are going to contaminate the product in the field, not in the handling facility, unless you're running cattle in your salad facility, which I hope you're not. There's another kind of special bacterium, Listeria monocytogenes, that we're familiar with, which is a soil-borne microorganism. Certainly does contaminate products in the field. It's a natural inhabitant of agricultural soils. It's on dust, it's in the air, it gets on our hands. It's everywhere, but it's at very low levels. The number of cells, the number of spores is rather low. Low enough that it's not likely to be a health threat. And it becomes a health threat when it becomes established, grows to high levels, and then those large numbers contaminate the products. And again, that is likely to happen in our handling facilities, not in the field. So the initial contamination might happen in the field or might happen in a handling facility. But the high levels of Listeria that are likely to make people sick are due to mishandling or poor sanitation practices in a handling facility typically. So I think that we need to start with an understanding of where these come from and what we actually have leverage on in our post-harvest handling, what we can and can't do about these things. In terms of human contact contamination with viruses, with Staphylococcus and the like, it's a very low incidence. Most people, most of the time, are not shedding these viruses. It's an arbitrary distribution. I think it would be very difficult to model the likelihood of an individual shedding these viruses who is carrying hepatitis A, who is carrying norovirus, who is not. There certainly are symptoms, but there are also symptomless carriers of these things. So it might be a difficult prospect to try to model the potential, the incidence, the frequency of contamination with these viruses. Prevention is going to depend primarily on exclusion of people who are carrying these viruses. So it's going to be an awareness on the part of the supervisors to watch people and make a judgment. Are they healthy, or they seem not to be healthy? And if they seem not to be healthy, then I need to talk to them and figure out why. So there may not be much opportunity for modeling here. You may disagree with this, and we'll have plenty of conversations about it. But these may not be our best modeling opportunities. Some of the major problems or causes of foodborne illness that we hear about, the E. coli, EHEX, and Salmonella and the like, as I said, are probably the initial contamination is likely to happen in the field. And Jim presented some data that show that, yes, these things do exist in the field. You can find them. Contamination does happen. It's at a very low incidence. It's difficult to find them through testing. And yet we know they're there. We know they're there at a low incidence, and we know that they come into our facilities. And furthermore, we know that we cannot thoroughly decontaminate the contaminated product that arrives at our front door of these facilities, or back door, rather, of these facilities. The best that we can do is that we can prevent cross-contamination and amplification. And I think that a lot of times when we get foodborne illness outbreaks, certainly associated with bagged salads, it's because we have failed to prevent cross-contamination and amplification in our handling facilities. And one of the ways in which we can do this is through water. Water is a very good dispersal agent. If you put bacteria or viruses in water, it'll spread throughout the water, and anything the water contacts it can potentially contaminate. So we may have major sources of cross-contamination associated with our foodborne illness outbreaks. I think this is probably the case. There are many variables to consider because there are diverse handling systems, harvesting systems, cooling systems, transportation, and certainly wash systems. There are a lot of variables. And I'm not going to spend a lot of time here talking about those details. But there are opportunities for measurement and process control at the wash step. A lot of things are difficult to manage. We don't have a lot of process control over sick workers, healthy workers. But wash water and the potential for cross-contamination is a point where we do have an opportunity to measure and to control that process. There are a lot of measurements going on. Taylor Fresh Foods is doing 1 million a day, 1 million a second, 1 million a millisecond. They're doing a lot of them. But it's also true that other operators throughout the produce industry are measuring lots of things. And in many cases, I think people that have been in the industry for a while will tell you, in many cases, they're not doing a very good job. They don't fully understand what they're measuring. They don't understand the analytical tools that they're using. They may fail to properly calibrate them. They may fail to properly clean the probes. And many times they think they're measuring something when they're measuring something else. They tell you that they're measuring chlorine concentration, but in fact they're using pH test strips and writing that down as chlorine concentration. The produce industry is made up of a lot of uneducated people. And so in many cases, you've got a field worker who's a good worker, consistent, pretty bright, and so you put him in charge of these measurements. You elevate them and say, well, you're part of quality control now, you're measuring this. But they don't necessarily have the background or the training to know what they're doing. And so we have a lot of measurements, we have a lot of data available to us. Some of that data is unreliable. And so one of the challenges in modeling is going to be able to separate out what is reliable from what is unreliable. We can't just take numbers that are given to us and base conclusions on those numbers. We need to have some understanding of how those numbers were generated, where they came from, and what they mean. Nevertheless, I think that in this case, in cross-contamination through water, this might be a prime opportunity for modeling. I really think there's an opportunity because we have measurements, because we have process control, and because we know this is a critical control point. This is where a lot of our problems happen. Listeria is another unique situation because, as I said, listeria is going to be constantly coming into a facility, either on the products or in dust, in the air, on our shoes, whatever. Not enough of it to necessarily cause a problem, but it can become established and start to grow and reach high levels. Listeria will grow any place that is consistently wet, whether it's warm or cold. So it is almost unique among human pathogens in that it will grow at refrigeration temperatures. It grows quite nicely at 2 degrees Celsius. Doesn't grow very fast, but it will grow and over time can reach high population levels. And so in the case of Listeria, there's a very low probability of it being in any given place, which would be the case for any of these pathogens. But in Listeria, we have some idea where it's likely to be. It's not likely to grow to high levels on a conveyor belt, where product is passing over it constantly, where it's getting cleaned and sanitized on a daily basis at least. It's not likely to become established and grow to high levels there because that's a surface that's being used and cleaned all the time. In places like drains, drip pans, maybe some flume systems, cooling units, things like that, places that are rarely touched, that are not often cleaned and sanitized, that are difficult to access, but that are wet, maybe cold, maybe not cold. That's where Listeria is likely to grow. And so in a Listeria exclusion and detection program, we do have the ability to focus where we're looking. So that's an instance where testing might make sense. Jim was talking about the futility of testing for pathogens in the field. To some degree, testing for pathogens in a processing facility is equally futile, I think, because the probability of the presence of these pathogens is very low. Listeria is different because we can focus where our testing is going to be, and so we can gather data. There are very rapid detection methods. There's a company called Sample 6 out of Boston that has a phage-based system for detecting Listeria. They say it takes about 3 to 4 hours to get confirmation of it, which is close enough to real time that's actually kind of useful. And so consequently, there may be modeling opportunities here for looking at Listeria, the probability of its presence, the distribution, and such. I'm not sure I would consider this to be the highest priority for modeling, only because we kind of already know a good bit about it. We kind of know where it's likely to be. We know how to find it. We know how to test for it. We know what to do about it if we find it. But this is an area where we can have the information necessary to model if that becomes interesting to us. Let me talk a little bit more about water cross-contamination, because to my mind, this would be a high priority in a produce handling facility. Most fresh produce is washed at some point during its handling. And many people in the industry and outside the industry believe that it's being washed to clean it. This is a sanitation process. Let me assure you, it is not a sanitation process. We wash produce for cosmetic reasons. We wash it to make it look nice, to get dirt and dust and whatever's on it off of it, to make it nice and shiny. We may do it to remove undesirable substances, soil, in the case of lettuce, the latex that might leak out when it gets cut and such. We might want to reduce the initial microbial load, although there are a lot of data available on reducing initial microbial load, and a lot of those data suggest that reducing the initial microbial load lasts for a couple of days. Within a couple of days, microbial load is right back where it would have been if you hadn't washed it. It actually doesn't make much difference. There are a fair number of data that suggest that the initial microbial load doesn't have too much to do with quality or safety or shelf life or anything else. To make it safer, I'm not so sure about that. I think that anytime we touch fruits and vegetables with water, we have an opportunity to contaminate it, not an opportunity to decontaminate it. And so we need to be very judicious in our use of water. Water is used in all kinds of areas in the fruit and vegetable industry, in dump tanks, flumes, spray bars, huller shellers, lots of different places water is used. And operators are not always cognizant of it. They may be cognizant in a dump tank of, oh boy, I better sanitize that water in my tomato dump tank. But other uses of water, they don't think about the fact that, oh, the water itself could be contaminating my product. So we don't always pay the attention that we should. Here's a famous one, using a nice wet rag to clean tomatoes or avocados or whatever it is you're doing. I just got an email last week from some endive growers where, well, sometimes we get a little dust and dirt on them, so we use a towel to wipe that off. Well, that towel is a beautiful vehicle for cross-contamination. Whatever gets on that towel is now you're putting it on everything. So these are the kinds of issues in post-harvest handling and in the use of water. So what I like to say, just because I like to stir people up, is that washing fresh produce is not an opportunity to clean it. It's an opportunity to contaminate it. Water disperses the microorganisms. And here's a key point. Once contaminated, the fruits and vegetables can transfer them to the water. The water can contaminate them, and we cannot decontaminate them. Other than thoroughly cooking fruits and vegetables, we have no effective method to thoroughly decontaminate them. We can wash them in chlorine, in ozone, in peroxyacetic acid. We can expose them to ultraviolet irradiation. We can do all of those things, and we can get a couple of log reduction in the microbial loads. But if we start out with 10 to the 5th salmonella cells, we can get them down to 10 to the 3rd maybe even 10 to the 2nd. 100 salmonella cells can make you plenty sick, depending on the serotype. And furthermore, if that product gets warm for a short period of time, those 100 cells can become 1,000 and 10,000 cells in a relatively short time. So that we cannot, washing, none of these processes will sterilize it. They're not kill steps. They can at best reduce the levels somewhat. Now, for the case of potatoes and artichokes and things like that, yeah, we're going to cook them. So we don't have too many foodborne illness outbreaks associated with those. But for many other fruits and vegetables, we're not likely to cook them. And so we cannot rely on that. So I said you can't decontaminate them with chlorine or ozone, irradiation, peroxyacetic acid, or anything else. And that's exactly what I mean. That is absolutely true. And that's a bit of a revelation to a lot of people. It's like, whoa, really? Isn't that strange? But if you think about it for a moment, if we had a process for decontaminating our produce by eliminating human pathogens on them, we would not have any need for good agricultural practices, good manufacturing practices, HACCP, audits. We wouldn't need them, would we? We just have this process and we decontaminate them. The fact that we need those programs ought to tell you we can't decontaminate this stuff. So don't believe anybody who tells you they can. And there are plenty of companies that will tell you we get a 5-log reduction with our process. We have a sanitizer that will give a 5-log reduction. Well, yes, you do. If you take bacteria and you suspend them in water and you put the sanitizer in that water, you can get a 5-log reduction. But you're not getting a 5-log reduction on lettuce or tomatoes or spinach or cabbage or anything else. So don't believe them. Just a few examples of this. These are tomatoes washed. This is old work from a long time ago. Tomatoes washed with chlorinated water. And it started out with Salmonella Montevideo at about 5 logs per square centimeter on these tomatoes. And as you can see, you wash with pure water, you get a slight reduction. You put some chlorine in, you get a log, maybe just over a log reduction. I think we started with 10 to the 6th, I believe. So just pure water will give you about a log reduction, a little over a log reduction. You put chlorine in, you get another hit. You start adding more chlorine, you get a little more reduction, but it's sort of asymptotic. You reach a point where more chlorine isn't giving you any boost at all. There's still, in this case, 1,000 salmonella cells per square centimeter on those tomatoes. Well, why is that? What's happening? And you know, our best ideas about what's happening is that these microorganisms, of course, as we know, they're very small. And so even a tomato that looks like a nice, smooth, waxy surface to our eyes, if you're down at the 1 nanometer level, it looks like the Grand Canyon. There are lenticels and cracks and breaks and openings and all kinds of things. Lots of little hydrophobic niches where these microorganisms can hide. And so our aqueous sanitizers cannot contact them. And if they cannot contact them, they cannot kill them. Some of them can become internalized. They can actually get sucked in through the stem scar or elsewhere during washing processes, or through breaks and cracks, where they actually get in between the cells of the plant tissues. And again, the sanitizers cannot reach them. And so for these reasons, none of our sanitizers are fully effective at decontaminating them. And there are a lot of data that show this kind of thing. This is a stomate of lettuce, and I've seen lots of these micrographs, and it does seem like the bacteria preferentially like or attach to these natural openings and breaks and cracks. That tends to be where you find them aggregating, and it's maybe because these have been washed, and so those are the ones that you can't get rid of. But there are lots of places where they do attach, and we cannot seem to remove them. I mentioned that they can become internalized. Water can do a good job of that. And this is a case of tomatoes where if you put warm tomatoes from the field and you put it in cold water, the air in the intercellular spaces is cold, and the air in the intercellular spaces is warm, and when it contacts the cold water, the temperature of that air goes down and the air shrinks as the air gets colder. And as it does that, it then will suck in the water because the volume decreases as the air gets colder, pulls in, you get a pressure gradient, and it pulls the water in. If there are any living microorganisms in that water, they're now inside the tomato. And this was work that was done by plant pathologists at the University of Florida because they were concerned about decay organisms, but it's equally true with salmonella or other pathogens that may be in the water. So now there's a standard in the tomato industry that the water should be at least 9 degrees Fahrenheit warmer than the tomatoes, which eliminates the use of the dump tank as a cooling step. It's just a washing step now. How effective that is hard to say. And this is just a little dye study showing you that, in fact, that water does infiltrate rather deeply into the tomatoes. So once you get salmonella in here, there's nothing you can wash it with that's going to get rid of it. There's really not much you can do. So if water sanitizers don't decontaminate the produce, well, why do we use them? What's the point of sanitizing the water? And the point is to prevent cross-contamination. That's why we put sanitizers in the water. Clean water will give us a log to 2 log reduction of the microbes on our fruits and vegetables. Adding a sanitizer will give us a little more of a bump, but not a whole lot more. Jim's going to tell me otherwise because he's got a magic one. But the main point of these sanitizers is not to reduce the microload on the produce. It is not to decontaminate the produce. It is to make sure that the water itself does not become a medium of cross-contamination, a vector of cross-contamination. Prevention is really what this is about. So wash water sanitizers can prevent contamination if they're used properly, but they do not reverse contamination. If we bring in spinach that has E. coli 15787 on it, that spinach is going to have E. coli 15787 on it going out the door when we're done processing it. We cannot prevent that from happening. But it's a rare event, as Jim's data showed earlier. It's a rare event that spinach has this pathogen. And our task is to make sure that that one plant in 50,000, or one plant in 5,000 approximately, that might have it, does not contaminate every other plant that goes through our washing system. So that's what our sanitizers are for. Okay, let's talk about listeria a little more. I think we kind of went through this. Yeah, I think we went through that, so we don't really need to talk about that in too much more detail. Okay, can we turn off the live feed? Who's in charge of that? Can we turn that off now? Okay, this is a slide that Jim allowed me to use.